All right, so we have a few more important points to make about meiosis. So we said meiosis is reduction division. We're reducing the chromosome number so that's going from diploid to haploid. We're producing typically gametes, egg cells or sperm cells, by this process of meiosis. And that we're producing 4 daughter cells, and these daughter cells do not look exactly alike. In mitosis, we do produce daughter cells that are exactly like the parent cell, and every daughter cell that's produced is exactly alike as well. There's no difference there. But in meiosis, there's a lot of genetic diversity that has been introduced. And so this is achieved through what we call crossing over and independent assortment. And these happen during different times in meiosis. So one's during prophase 1 and the other one's during metaphase 1. So let's look at each of these. So crossing over happens during prophase 1 of meiosis 1. During this time we have homologous chromosomes. So we said homologous chromosomes are chromosomes that look the same and they have the same genes within them. But there could be slight differences in how the information is encoded within the genes. So within this image we have kind of this darker orange here showing a chromosome that may have been inherited from the father's side of that individual, and the lighter yellow maybe from the mother's side. And we're looking at 2 genes. So we have a gene for whatever character is controlled by A and another one for B. So we're showing that even though the gene is located in the same area of the chromosome for the dad's chromosome and the mom's chromosome, how the information is encoded could be slightly different. So that's why we have a capital A versus a lowercase a. We said this could be a gene for eye color, and maybe that gene encodes the information on how to make brown eyes. Well, this one over here is still the gene for eye color in that location on the chromosome, but it could be how to produce blue pigmentation for blue eyes. So there's a slight difference that can occur that you could inherit from each of your parents. So what happens during prophase 1 is these homologous chromosomes find each other and they pair up. And when they pair up, they can have portions of their chromosomes swap over. So they kind of overlap each other, and they'll swap portions of each other's chromosomes. So they'll actually detach a piece of the chromosome, and the same area will detach from the other chromosome, and they'll swap between them. They'll say, here, you take this piece, and I'll take your piece. So the same genes are swapped out. So each one now in this image has still the gene for B, but now what was originally on the mom's chromosome is on the dad's. And what was on the dad's is on the mom's. Now notice this other gene was not affected at all. So crossing over is something that's completely random. We don't know where it will happen, but typically 3 crossing over events happen every time homologous pairs get together. And it could occur with these upper arms as well. There could be crossing over happening. So they swap portions of their chromosomes out between them. So this is crossing over, and this is how some of that genetic diversity is introduced, because an individual, when we separate these chromosomes out into daughter cells, are no longer just getting a chromosome that's all the paternal information or all the maternal information. They're now getting a chromosome that's kind of a mix of paternal and maternal information. The other piece of information that contributes to genetic diversity in the daughter cells produced by meiosis is what we call independent assortment. So this occurs during metaphase 1 of meiosis 1. So this is after crossing over happens. The homologous chromosomes are being pulled by spindle fibers, and they start to line up in the middle of the cell along that imaginary line that we call the metaphase plate. Now, how those homologous chromosomes line up is completely random, right? It has nothing to do with other sets of homologous chromosomes. Each set will line up independently of the other sets. So it can lead to a wide variety of different possible combinations of maternal and paternal information in the daughter cells, especially if you have a large number of chromosomes. So in this example, we're showing a cell with just 2 different types of chromosomes. So once again, we're looking at a large chromosome and a small chromosome. But think about what can happen in humans with 23 different sets of chromosomes. So there would be 23 of these all lined up in the cell. So we're showing 2 different possibilities that can occur with these 2 sets of chromosomes. So we have option A on the left and option B on the right. On the left, you see they're showing the maternal chromosome in red and the paternal one in purple. So in this case, we could have the large chromosome from the mom lining up right above the small chromosome from the mom. Over in option B, it's switched so that the large chromosome from mom is right above the small chromosome from dad. So how they line up, we said, completely random, right? Each homologous pair will line up irregardless of the other pair. So think about what's going to happen here, right? These homologous chromosomes are going to be separated. So we're going to divide this in half here, right? Because these spindle fibers are reaching out and making connections, and they're going to pull the chromosomes to opposite sides, right? So these are going in opposite directions. And then they get packaged up into their daughter cells. So once again, in option B, we're going to separate those and pull them out into each of their daughter cells. So now we look at these haploid cells, and this one has all maternal information. This one has all paternal information. This one's a combination of mom and dad, and this one's a combination of mom and dad, but the combination's different, right? As we go through meiosis 2, we're separating out the sister chromatids. So once again, we're going to divide this in half. We'll draw the line down the middle here to show how this is going to happen, because they're skipping some of the stages here, right? So now you can see we're going to produce 2 daughter cells from that one that are still going to have all maternal information. But now we just have that one copy of the large chromosome and one copy of the small. These 2 will have all paternal information, and these will still be a combination of maternal and paternal. But they're different, right? Large red over here, large purple over here. So in option A, we have a distinct, each of the daughter cells formed here are just going to be all maternal information or all paternal information. So you ever wonder why you look like your grandfather, maybe more so than your grandmother, right? Versus these are all maternal and paternal combined together. So there's different possibilities that can happen, and this is what's called independent assortment. So once again, think about what would happen if you had 23 pairs of homologous chromosomes lined up, and all the possibilities for maternal and paternal combinations as they all 23 pairs line up, and then they're divided out into daughter cells, right? So this is why, brothers and sisters don't always look very much alike, because there's so many different possible combinations that can be produced when we're going through this process of meiosis. Now, this doesn't even take into account crossing over, right? That's not even shown here, the random crossing over events that could have happened before independent assortment, right? But either way, crossing over and independent assortment kind of ensure that the daughter cells that are produced by meiosis are very distinct one from another. And this introduces genetic variability into those gametes.